right? We'll start with learning objective 1. So we'll try to look at the first 2 learning objectives here, and then a review. So understand the differences between chemical and electrical synapses, okay? So let's sort of describe exactly what a synapse is. A synapse, we said, is the functional connection between a neuron and another cell. And the reason that we say a neuron and another cell is that it doesn't have to necessarily be another neuron. Most of the time it is another neuron, but it can also be things like muscle, smooth muscle, cardiac muscle, or even skeletal muscle cells, as well as glands, right? So all of the glands in our body can also receive innervation from neurons. So a synapse is the communication, right? In other words, this is the point where one neuron can speak to another cell, whether that be another neuron, smooth muscle, cardiac muscle, or glandular cells. This junction, specifically for muscle, is going to be called the neuromuscular junction. For other cells, it's going to be called purely a synaptic junction. Now, if one neuron signals another neuron, then we can think about the neurons in series. The first neuron in that series is going to be called the presynaptic neuron, and the second neuron in that series is going to be called the postsynaptic neuron. And so a presynaptic neuron can signal specifically onto the proximal parts of that additional neuron or cell, right? The soma, the cell body, right, which is the proximal bit of the neuron. And that area is going to be receiving input signals from a lot of other different neurons. And we kind of illustrated that before. So the presynaptic neuron can signal by way of its axon terminal onto the cell body of the postsynaptic neuron, okay? It can also synapse onto some other areas of the neuron as well. And this is where we have different types of synapses. So we can have the axon terminal synapsing onto the dendrites, which is the most common synapse. We can have the axon terminal synapsing on to the soma, right, which is another type of synapse, or we can have the axon terminal synapsing onto the axons. So we can have an axodendritic synapse, an axosomatic synapse, or an axoaxonic synapse, depending on specifically where that presynaptic neuron will synapse. All right. Now, as you mentioned earlier, most of the synapses that we encounter will be axodendritic, and that is going to be most of what we speak about as far as what we look at going forward. Now, synapses can be electrical or chemical, And so the main examples are electrical synapses are going to happen via gap junctions. So this is where this cell can speak or communicate to this cell via these channels in the membrane, which are called gap junctions. And so the electrical impulses can go right through from one cell to the other, going through these gap junctions. Okay. Chemical synapses, on the other hand, we're going to have to convert that electrical energy into a chemical. And this is where we have neurotransmitters involved. So a neurotransmitter is a chemical signaling molecule or communication molecule that will be released at the axon terminal. So if you think about this being the proximal end of the neuron, this being the axon, right? So up here is where we have the dendrites. So the dendrites are sticking out. The soma, or the cell body, which is the bulk of the neuron here. The axon hillock being this neck region. The axon being the entire projection, and then the axon terminal down here. So this is our presynaptic neuron. This guy is our postsynaptic neuron. And then this will be considered the synapse, so the area where these two neurons speak to each other. And so if you look more closely at what is happening at this synapse, we have to have the electrical energy, which is the action potential. So the action potential goes down the length of the axon, and then when it gets to the terminal, it has to be converted into chemical energy. And then that chemical energy can cross the junction and be perceived or be taken up onto the postsynaptic neuron, and then that chemical energy will be reconverted to electrical energy via the graded potential, and then that can be converted to a new action potential on this neuron and go down the length of the adjacent axon. So this is how we communicate from one axon or one neuron to the other. Electrical energy converted to chemical energy at the axon terminal, that chemical energy crossing the synapse, being perceived here as electrical energy again. That can be perceived as a graded potential, which we said is required to result in an action potential. And then the action potential, which is started at the axon hillock, will propagate down the axon, and then that will happen again and again and again. And this is how we can have signals being sent over long distances by way of the chemical and electrical synapses. All right, so looking a little bit more closely at the features of an electrical synapse, it's going to be happening by way of gap junctions. Some are going to be between neurons and glial cells. So glial cells are essentially the supporting cells. We'll look at those in the next chapter, but neurons have lots of supporting cells around them, and so they can also communicate onto those supporting cells as well. Some of the functions of the electrical synapses is for rapid communication. So notice when we talked about the electrical synapse being converted to a chemical synapse in order to cross the synaptic junction, that's going to take some time. That's going to slow down the rate of this communication. An electrical synapse, on the other hand, is going to be really rapid, really fast. And so if it's able to just go right through this gap junction, it's not going to be transferred to a chemical and then back to an electrical, which is going to take up some time. So if we look at electrical synapses, they're going to be much more rapid in their communication than chemical synapses are. Another feature, ions or second messengers are typically involved. Also, we can have bidirectional flow. So we can have this neuron send signals to this neuron, and then we can have communication going in the opposite direction. So this neuron can likewise send signals back across to this neuron. So there can be bidirectional flow of communication. For electrical synapses, for chemical synapses, that is not the case. For chemical synapses, there's only one flow of communication. So dendrites to soma, to axon hillock, to axon terminal. We cannot have any flow of communication in the opposite direction. For electrical synapses, we can have excitation and inhibition happening at the exact same synapse. So those can be summed together to see which types of signals are stronger, but we can have both types of events happening. And then some of the areas in the body where we see electrical synapses are going to be the retina, the cortex of the brain, which is the outer part of the brain. That's going to control actions like breathing, the hypothalamus as well. So very few areas in the body. This is a very rare phenomenon in our body. Most of our synapses are chemical, and then a very few areas in the body do we have electrical synapses. In fact, early scientists did not understand electrical synapses very well and thought that they were only found in the developing brain. And then we moved on to realize that there are some instances in the body where we do see electrical synapses persisting even into adulthood. So some of those examples are listed here. Again, retina, the cortex of the brainstem, and then the hypothalamus. Okay. Now, electrical synapses, again, very rare. Chemical synapses, on the other hand, are much more common and are going to be the focal point for what we go on to discuss. So chemical synapses are requiring two neurons, a presynaptic and a postsynaptic. It's going to be the chemical energy or the chemical transfer is happening across the synaptic cleft. Chemical synapses are also unidirectional, so only one-way flow of this communication. Usually it's going to be from the axon terminals of one neuron to the dendrites of the adjacent neuron, so the axodendritic version or axodendritic type. And then some synapses, as we mentioned earlier, can take place at other points of the neuron. All right, so let's look at the various types of synapses that we can see. So here's our presynaptic neuron. Here's one example. Here's another example. Here is our postsynaptic neuron, right? Here's another example of that. So the axodendritic, this is where we go from the axon terminal to the dendrites, right? These are the dendrites, these spiky projections coming off of the soma. This is the most common type of synapse that we will see. The other examples are the axosomatic, where we have the synapse taking place from the axon terminal to the soma itself, right? Remember, soma is synonymous with cell body, which is the area of the neuron where we have the cytoplasm, the nucleus. So this is the axosomatic synapse. And then we've got the axoaxonic, where we go from one axon terminal also to the other axon terminal, right? So those are some of the other types of examples that we see: axosomatic, axoaxonic, but axodendritic is the most common that we will explore. Question here: what does it mean by the synaptic cleft or the point of it? So the synaptic cleft is this region. Okay, so going back here, we zoomed in on what's really happening on a microscopic level between this presynaptic neuron and the postsynaptic neuron. So the space between here is what we refer to as the synaptic cleft. Okay, on a big picture view of that, that would be this space between here. All right, other terms for that that area is the synaptic junction, right? So synaptic junction, synaptic cleft. So is it thicker on chemical synapses? So it's not only thicker, but it's completely different, right? This is a really good question. It is thicker, it's wider, because we're going from one neuron to the other, and that space is really wider. Here at the electrical synapse, there really is no space. Another reason that this happens much more rapidly than it does for the chemical synapse. So there's no space. The cells are pretty much adjoined by these gap junctions. So yes, we can think about the chemical synapses as being thicker or wider, right? In terms of the space where that chemical communication is happening. For the electrical synapse, there really is not a space. The signal is going right through that gap junction from one cell to the other. Okay. All right. Okay. Any other questions from that first objective, which is basically just describing the types of synapses that we see, also describing exactly what a synapse is, where it is, right? It's that junction of communication between two cells. Now, just to reiterate, the chemical synapse is the more important synapse. That is what we're going to look at in learning objective 2. The electrical synapse, I want you to know what it is. I want you to know how it differs from the electrical synapse, but we're not going to speak much in detail about the events that happen. But I do want you to know that it's faster. It requires gap junctions, okay? And it is exclusively electrical energy, and it is bidirectional. All right, for chemical synapses, it's not exclusively electrical. That action potential, which is electrical, has to be converted to a chemical first. That has to cross the chemical synaptic cleft, and then that can be reconverted on the adjacent neuron. So it's going to take a little bit more time. It's not as rapid as the electrical synapse was. Simply by way of all of the machinery that's required to make these neurotransmitters, which are the chemicals that we're referring to here, release them from the axon terminal, send them across the junction, bind to that postsynaptic neuron, and then convert that into a graded potential and then a new action potential. So notice how there's a lot more steps involved, and that is why it's going to take a little bit more time. That is also why it's only going to travel in one direction. Okay, so our second objective, to describe the communication across the chemical synapses, explain how neurotransmitters are released, and describe their actions after they are released. Okay, so let's look more in detail as far as the anatomy of the synapse. What are the players? What are the elements or components that are involved? So first off, we talked about the first one here, the presynaptic axon. Here's our presynaptic axon, right? A closer view of that. Here's what the big picture of that looks like, right? And this is the area that we're really zooming in on. So this is the area that we're really kind of focusing in on. So this is our presynaptic neuron. This is our postsynaptic neuron. This would be the synaptic cleft. What are the other players? The neurotransmitter containing vesicles. So there are small vesicles within the axon terminal that actually have the chemicals that we are referring to. This is why we call this a chemical synapse, because there are some tiny chemicals inside these vesicles, which are referred to as neurotransmitters, that are going to help transfer that electrical energy. What is that electrical energy? It's the action potential that's coming down this axon. So think about our action potential, which starts here, right? This is the axon hillock, where the action potential is initiated by way of the sodium channels, that we described. It's going to move down the length of the axon. This is all going to be considered an electrical transfer. When it gets to the axon terminal, we're going to convert that electrical energy into chemicals. So the neurotransmitters that are stored in these vesicles are going to be triggered to release their contents into this cleft. All right. These little chemicals or neurotransmitters will bind very uniquely onto receptors on the adjacent neuron. And then that will convert these chemicals into new electrical signals. And then those will be perceived as graded potentials on the additional neuron. And then a new action potential will be initiated. Okay, let's look at the other players here. The voltage-gated calcium channels. So that's these guys. These are what are going to trigger the release of these chemicals. How are they going to be initiated? A voltage change. So because they are voltage gated, when the action potential gets to the axon terminal, it's going to stimulate a membrane potential change. We know this. We know that an action potential, by definition, is a change in the membrane potential, which increases it up to positive 30. That in and of itself opens up calcium channels so that calcium can trigger the release of these neurochemicals or neurotransmitters, and they can be dumped into the synaptic cleft. All right, what are our other players? The synaptic cleft. So we describe this. It's just a space here. That is referred to as the synaptic cleft. Receptors, which are these players. What are they going to be doing? Binding the chemicals that are released in the junction, in the cleft. Enzymes. There are also enzymes that are bound onto the postsynaptic membrane. What is the purpose of the enzymes? They're going to help to degrade any leftover neurotransmitter that has not been bound to a receptor. Okay, and then the reuptake molecule, which is another player here. The reuptake molecule is actually on the membrane of the presynaptic neuron. And what is the function of the reuptake molecule? Just as the name implies, it's going to reuptake any additional neurotransmitter that has not been bound. That's going to be a really important part of clearing the junction after this signal has been transmitted. So the receptors here will bind this neurotransmitter that's been released into the cleft. The enzymes will degrade or break down any excess neurotransmitter that has not been bound, and then the reuptake molecule will sort of recycle any of those chemicals that have also not been used. So the enzyme and the reuptake molecule are two ways that we can clear the excess neurotransmitter from the junction or the cleft.